My name is Tessa Mlinar, and I am presenting on the effects of cell-specific optogenetic stimulation in the subthalamic nucleus for the treatment of depression. I chose to focus on this project because of the previous work being done in Dr. Yu's lab on deep brain stimulation for Parkinson's disease. Upon further research on this topic, it was made clear to me that depression is a common side effect suffered by those with Parkinson's disease, and the causes and treatments of PD-induced depression are unknown. We began this project to test if deep brain stimulation could be used for treating depression. In addition to the behavioral study conducted to test this theory, we also performed a tracing study to delineate the behavioral and motor subdivisions of the STN. In figure 1 here, you can see the viral expression seen in the STN, and then in B of figure 1, you can see the actual location of the STN. To perform the tracing study, CAMK2 was injected into the medial prefrontal cortex and allowed 4 weeks to be expressed. These brains were then harvested, sectioned, and imaged by a fluorescent microscope. To perform the behavioral study, we had 4 groups. We had 2 control groups that consisted of a naive control, which performed the depressive test but did not undergo chronic mild stress or surgery, and a saline control, which did undergo chronic mild stress, had sham surgery, and sham stimulation. The test group was split between Chronos and GFAP, where Chronos injections targeted the excitatory cells in the STN, and GFAP injections targeted the glia cells in the STN. The chronic mild stress protocol is a commonly used process for inducing depression in animals. It consists of exposing the animals to 3 or 4 random stressors throughout the day for 5 weeks. After CMS, the animals underwent a series of tests to both confirm depressive behavior and track alleviation of depression after deep brain stimulation. These tests included the forced swim test, Sucrose splash test, and Sucrose consumption test. Animals received 90 minutes of stimulation the day before testing and another 90 minutes on the day of the test. A preliminary study was performed to test the effectiveness of the chronic mild stress protocol. In this, we found a moderate decrease in sucrose preference, a sign of anhedonia, a clear decrease in swim mobility, and a slight decrease in grooming behavior. Once we confirmed that CMS was effective for us, we began our testing. One of the unique findings that we found was the trend seen in the animal's body weight here. That's in figure 2. At the beginning of this process, the average animal all weighed 240 grams, regardless of which test group they were in. But as the animals went through CMS, their weights continuously decreased, even though they were still growing and should have been gaining weight. And then here you can see that the naive control did continue to gain weight throughout the 5 weeks, as was expected. In the forced swim test, both GFAP and Chronos groups had an increase in swim mobility following DBS and a decrease in floating behavior. Floating behavior instead of swimming or climbing is a sign of depression in animals. The Sucrose splash test, that's where we observe the grooming behavior of animals after they are sprayed with a 10% sucrose solution. Having a low percent of time spent grooming is indicative of depression in animals. After stimulation, the GFAP and Chronos groups both increased their time spent grooming by 15%, and the control groups had no significant change. Finally, rats have a natural preference for sweet foods, so the sucrose consumption test checks for anhedonia experienced by depressed animals. At the end of CMS, the test groups had a low sucrose preference ranging between 55 and 75%, whereas naive control animals remained at a preference of around 90%. After 1 day of stimulation, the Chronos and GFAP both increased in preference by 31% and 17% respectively. This brought them up to the same value as the naive control group. After a 2nd day of stimulation, the test animals had even higher sucrose preferences than the naive control group. No difference was seen for the saline control after going through sham DBS, which helps confirm that stimulation is what caused this sudden increase in preference. In conclusion, this project was able to find that stimulation can reverse anhedonia symptoms, increase swim mobility, and overall decrease depressive symptoms. The research is still ongoing, but results so far are promising and provide support that deep brain stimulation could be used as a treatment for depression. Finally, I would like to thank the Summer Undergraduate Research Fellowship Program for funding this project, and Dr. Yu for her support, knowledge, and advice as I worked on this project. Thank you.